Good afternoon, everyone. Myself, Monica, from Department of Physics, Miranda House, University of Delhi. The topic of my talk today is the LAM wave-based acoustic devices for wireless sensing applications. First of all, I would like to thank the organizers for giving me this opportunity to present my work. Today I am going to present the work that we are doing for the fabrication of the LAM wave-based acoustic wave devices for wireless sensing. So to begin with, what a sensor? Sensor is nothing but a device that measures some physical, chemical, or the biological quantity, or which is there in its vicinity. So as a result of the presence of these quantities, some properties of the sensors do change, and we are able to measure those changes. And there is a transducer which converts it appropriately so that we are able to see the signal. So the good sensor should have high sensitivity, good selectivity, low detection limit, quick results, good linearity, high stability, repeatability, reproducibility, large shelf life, and low power consumption. So there are various kind of sensors on which people are working, like the semiconductor sensors, the most popular one, electrochemical sensors, SPR based sensors, gas chromatography based sensor, infrared spectroscopy based sensor, and also the SAW based sensors. So the focus of my work in this talk is the SAW based sensors. We are focusing on to the SAW based sensor because the sensors are of very small size. These are cost effective, and when you produce in the bulk, then the cost can be really brought down to a low level. The sensors have high sensitivity. They perform the wireless operation also. Wireless operation means like you can put the sensor in the far field zone, and you can get the signal in your computer or on your mobile phone also. These are low power consumption, room temperature operated sensors. So what are surface acoustic waves? Surface acoustic wave is nothing but some acoustical waves which are traveling onto the along the surface of a material, which exhibit the elasticity. This is the property of the material, that the waves, the mechanical waves, can travel onto the surface, and the amplitude is decaying exponentially with the depth onto the substrate. So there are different applications of the SAW bandpass filter. Is the one which allows only one single frequency to pass, and the frequency is inversely proportional to the acoustic wavelength. Acoustic wavelength is nothing but the periodicity of the interdigital transducer electrode, which is fabricated onto the piezoelectric substrate. So the SAW devices find variety of applications like in filters, delay lines, resonators, gas sensors, biosensors, pressure sensors, magnetic field sensors, and the UV photodetectors. So in the present work, since we want to focus the onto the sensing applications, so we have moved on to the LAM wave SAW devices. Now, what are LAM wave devices? LAM wave devices are when the entire SAW device, that is the piezoelectric substrate and the two transducers, are fabricated onto a membrane. So if you reduce the thickness of this substrate and you make the entire device onto a membrane like this, then the entire membrane propagates while the waves are propagating. So here we are having the LAM wave devices, and whenever an external signal comes, it causes perturbation with the surface waves which are there, and as a result of that, the frequency of operation of the device changes, and this can be related directly to the presence of the external signal, its quantity and concentration also. So amongst the piezoelectric layers, there are different layers which are used in the SAW devices, like lithium niobate, PZT, aluminum nitride, gallium nitride, and zinc oxide. All of them have their own advantages and disadvantages. Amongst them, we will prefer the zinc oxide because it is a very good sensing material because of its absorption and desorption properties. And then it is biocompatible also. We can easily fabricate it using number of techniques, and it is cost effective. But the challenge is that we can use it for the intermediate frequency regions only because of its low acoustic velocity. However, we are working towards increasing the velocity also by the lithographic capabilities and using the layered structure, where we can use the substrate which are having the higher acoustic velocity, and then the overall frequency of operation of the device can be enhanced. So these are different steps for the used for the fabrication of the surface acoustic wave devices. We take a passivated silicon wafer, we open a window from the back side, and then we do the wet etching so that the extra part where the window is opened is removed. Like this, a membrane is formed. Then onto the membrane, we deposit the zinc oxide thin film. On zinc oxide thin film, we do the IDT patterning, and then we do the further dry etching so that the thickness of this membrane is further reduced and we get the membrane thickness of appropriate size. Once the device is ready, we do the wafer dicing, then the mounting of the devices onto the header, then the final packaging. Then the using this package, we can use it onto the ZIG for the RF biasing, and then finally frequency analysis on the VNA. So like this, the complete device is fabricated and tested on the VNA. So the most important component is the zinc oxide thin film deposition, which we have deposited using the RF sputting technique, which is available in our lab. And these are the typical deposition parameters which are used for the growth of the zinc oxide thin film. Now we have optimized the zinc oxide thin film so that the SAW device could be fabricated on that, and the losses are as low as possible so that the acoustic losses reduce. So we have varied the pressure from 20 millitorr to 35 millitorr, and we have also varied the gas composition from oxygen contained in the argon oxygen ratio from the 60% to from 50% to 80%, and we find that a particular composition of the oxygen argon ratio of 60 to 40% and gas pressure of 30 millitorr gives a zinc oxide thin film which has low losses, the surface is not very rough, and the film is good for the fabrication of the SAW devices. Then we designed different masks. We had chosen 4 different kind of devices. I will be talking about these 4 devices later on. And then we have the 2 masks. One mask is for the fabrication of the IDTs. This is the layer 1, which is for IDT, and the layer 2, which is for the opening of the window from the back side. And these are the two, the 2 masks should be exactly positioned in such a way that the IDT faces exactly on the top of the membrane. So these are the typical specification of the fabricated zinc oxide, silicon dioxide, silicon lamb wave device. The thickness of different layers is kept as zinc oxide is 5 micrometer, silicon dioxide is 2 micrometer, and silicon membrane is kept at 37 micron. So we have the different devices that we fabricated, D1, D2, D3, D4, which are having the different acoustic wavelengths like 20 micrometer, 40 micrometer, 60 micrometer, and the 80 micrometer. So based on that, the devices were fabricated, and these are the response of the 4 devices. Now, in the first device, that is D1, where the lambda is 20 micrometer, but the thickness...
Madam, please conclude your work in 3 minutes.
H is very high, so the H by lambda is very high, and we do not get any lamb wave mode. But for the other devices, when the H by lambda becomes equal to approximately equal to 1, or when it becomes much less than 1, then we get the very good lamb wave modes. So these were matched with the theoretically obtained results, and we want the 100% matching. Then the fabricated device were utilized for the low pressure sensing, and we got the very high sensitivity of around 275 ppm per psi in the range of 0 to 10 psi. The important thing is these devices are not available in the market, and we have to work on this. Then we utilize these devices for the NOx sensing also, and we find when we compare them with the relay wave based devices, then the sensitivity is about 2 orders higher. Then we utilize these devices for the UV ray sensing also, and we find that the devices are highly sensitive towards the UV radiations also, specifically the higher modes, which show very high sensitivity, and the sensitivity value is much higher compared to those reported in the literature. Then the main aim was for the detection of the chemical warfare agents. We utilize these devices for detection of chemical warfare agents. We had chosen 3 sensing layers: zinc oxide, tin oxide, and titanium dioxide. We utilize them for the DMMP and the DBS, These are the two simulant for the chemical warfare agents. All of them showed some sensitivity. Then we did the polar plot for the sensing devices with the 3 sensing, and we got the different patterns for DMMP as well as the DBS. And as the concentration is increasing, then the area of these patterns is increasing. So we can detect, using this E-nose, we can detect the presence of different chemical warfare agents in the warfare. We also studied the selectivity, reproducibility of these devices, and we find the devices were working very well. And when fabricated in different batches, they were showing us the similar results. So in conclusion, the lamp wave devices, which were fabricated using the zinc oxide silicon dioxide silicon structure, they have been fabricated for the frequencies less than 500 megahertz. And these devices can be utilized for efficient detection of pressure, chemical warfare agents, and the UV radiation. So I would like to acknowledge my collaborators and my student support. My collaborators are Professor Arjit Chaudhary, Anjali Sharma, Lokesh Rana, Dima, Malika Verma, and Kajal Jinder. And my students, Manisha, Ajay, Jatinder, Babita, Satyam, and Shweta. So these are some photographs of the instruments which are present in our lab. And thank you very much.
I am Mukesh Kumar, working as assistant professor of
Yes.
physics in Government College Sector 9. I want to thank the organizing committee for inviting me for presenting my work. And wait a minute. Today's topic of presentation is synthesis and characterization of superparamagnetic NFO nanoparticles using hydrothermal method. These are the outlines of my work. Introduction of ferrite, experimental method and characterization technique used, conclusion. Basic introduction of ferrites. Ferrites are generally divided in two types on the basis of their magnetic behavior. Soft ferrite and... Hanji sir?
Your slides are
Hanji sir.
okay. Your slides are not visible.
Wait sir. I am not able to listen you.
Yes, yes.
What happens sir?
Slides are visible, Mukesh.
Now visible sir?
Yes, yes,
Make
yes.
Make it full screen.
I make it full screen. Now it is full screen, I think.
Yeah, you continue.
Ferrites are divided in two types on the basis of magnetic behavior, soft ferrites and hard ferrites. And on the basis of their crystal structure, they are divided in three types. Cubic ferrite having general formula MFe2O4. Garnets having general formula A3B5O12. Here A, B, M are magnetic particles. Sorry, metal ions. Hexagonal. Third one is hexagonal B12O19. I work on the cubic ferrites and their structure is spinal. That's why they are also called spinal ferrites. In their crystal structure in itself, there is 32 oxygen ions, which creates octahedral site and tetrahedral sites. Octahedral site also called D site and tetrahedral site called A sites. On the basis of presence of the metal ion on these A and B side, the cubic ferrite are further divided in normal spinal, inverse spinal, and partial spinal. If 8M, in this formula, there are 8M positive ions are present on A site only, and 16 Fe3 positive ions are present on B site, then this structure is called normal spinal. And if out of these 16 Fe3 positive ions, 8 are present at A site and 8 are present at B site, and remaining 8 M dipositive ions are present at B site, then this is called inverse spinal structure. And if there is मान लो 8 है, 8 में से 2 हमारे बी साइड पे चले गए डाई पॉजिटिव एंड 6 यहाँ पे ए पे हैं, ऐसे ही 16 में से 14 इधर हैं, 2 उधर चले गए, तो इसको हम पार्शियल स्पाइनल बोलते हैं. तो इस तरीके से 3 स्ट्रक्चर में डिवाइडेड रहता है. Why I use the ferrites? because ferrites have many applications in nanoelectronics, sensor, catalyst, biomedical, and batteries, etc. And these applications of ferrite depend on their magnetic and electrical properties. And these properties are further depend on the size and morphology of the nanoparticles. Nanostructure growth of ferrite depend on many factors like pH, temperature, reaction time, solvent, surfactant, sintering temperature, and method of synthesis. And by changing these parameters, we can easily tune their properties according for a required application. That's why the ferrites are used largely. Synthesis method and characterization technique used. I use hydrothermal method for the synthesis, and characterization techniques used are XRD, Raman spectroscopy for crystal structure formation, physical property measurement system for magnetic behavior, SEM, and TEM for their morphology. This is the schematic for hydrothermal method. Here, first we take the nickel salt and iron salt in 1:2 molar ratio. And after stirring, we add the reducing agent for maintaining the pH value. I use here two type of reducing agent, NaOH and NaBH4. And after stirring half an hour, we put the solution in the autoclave for some time T and at some temperature T. I fix here time T 12 hours, but vary the temperature of this oven. And after 12 hours, we stop the oven, and after cooling it to the room temperature, we get a solution. And after washing and drying these, we get the final powder. Here we synthesize NF4 nanoparticles by varying synthesis temperature with NaBH4 as a reducing agent, then we called it N series, and with NaOH as a reducing agent, then we called it T series. These are the precursor used, and synthesis time, annealing temperature, annealing time. Here precursor used are ferric nitrate, nonahydrate, nickel nitrate, hexahydrate, and reducing agent we use NaBH4. And synthesis temperature variation is 80 degree, 100, 120, 140, 160, 170, 200, 220, and 240. And naming is also with these temperatures. Synthesis time is 12 hour, annealing temperature 80 degrees Celsius, annealing time overnight in oven. In the second series,
Gathered here for the second international conference, which was organized by the Sant Kabir Institute of Technology and the Management. And my research topic is the synthesis of silver nanoparticles using the plumeria plants and larvacidal activity against the malaria vector. This is my short outline of my today's presentation. Here I am presenting the abstract and introduction, objective of my research, then outstanding feature of synthesized material, then preparation of AgNPS particles with the help
Sorry to interrupt you, Dr. Nanda. You can make in slideshow full screen.
It is in a full screen, sir, actually.
Dr. Nankishore, you can switch on your video as well. It is required.
Yeah.
Okay. Just a minute, sir. I think, may I visible, sir?
Yeah, video is okay. You can share your screen also.
Yes. Is it visible, sir? Hello?
Yeah, but you can make in slideshow, sir. You can make in slideshow full screen.
Yes. Okay. This is the abstract of my today's presentation. Here I was synthesized the nanoparticles, that is the silver nanoparticle, with the help of plant material which is easily available in our local areas. Then after the reduction, there is a formation of a silver nanoparticles which was characterized with the help of different characterization techniques. And we also done the antimicrobial activities. This is a research outline. This is the introduction of the today's presentation, as we are selected the silver nanoparticles.
Dr. Nandakishore, sorry, slides are not moving.
Oh,
Now we are in abstract only.
okay.
Actually, even that is not in full screen mode.
Sir, may I show like this?
Yeah, okay.
Then it is more flexible, I think.
Yeah, okay, you can.
Okay, thank you. As I've selected only the silver nanoparticle instead of other, because silver nanoparticle can use the very good antimicrobial activity, and it is now used in the nanoscience and the nanotechnology. And here we are using the simple biological method instead of the chemical method, because this is the simple, rapid, non-toxic, and we use the green approach. That's why we are selected only the biological method. Then we also inhibit the growth of bacteria with the help of silver nanoparticles. And one thing I was done that synthesized material can be employed into the society nearby areas of the my college. This is the objective of my research. Here we are synthesize the nanoparticles with the help of the plumeria plant. In local language we call it as the chafa extract. And we also done the physicochemical characterization of the synthesized material, and we done the larvacidal activity. And later on the synthesized material can be employed into the society, so it have the social applications. Here is the methodology. As we know that right now we are facing the number of problems with the mosquito diseases like Zika, fever, malaria, dengue, chikungunya, and that all type of the diseases are spread due to only the mosquitoes. And if we break the life cycle of the mosquito, ultimately the spreading of the diseases like this can be stopped. So this is the methodology of the work. Then this is the outstanding feature of my work. We are employed this material into the social application, and we got the appreciation from the municipal corporation of the Malegaon and the Nashik. And later on, we are using the green solvent. That is, we are using the water. That's why we call it as a green solvent. Then it is the eco-friendly and having the high thermostability, which was characterized with the help of the thermal gravimetric analysis. Then this is a preparation of the synthesized material. We are taken the plumeria plant, that is the Jaffa plant latex, white color latex, which are mixed with the water and the silver nitrate. After stirring, there is a formation of the silver nanoparticles, and we are characterized this with the different characterization techniques. Later on, this is an experimental setup of the synthesized material. This is the actual picture of the plumeria plant. Later experimental section, we are done the scientific survey by my research group. My MSc student collected the larvae from the different area of the city and nearby areas of the Malegaon, and we collected this sample with the help of the zoology department. Then these are the appreciation by the municipal corporation of the Malegaon and nearby villages, that our research group done a very good survey on the mosquito larvae and its control in nearby areas. That is the science for the society. Here are the FTR analysis of the synthesized material and the UV visible spectra of the silver nitrate. These are the morphological analysis of the synthesized material. As we can see, the synthesized silver nanoparticle consists of the spherical shape with the average size of the 70 nanometer. These are the anti-larvacidal activity of the synthesized material. As we are using the silver particles, which are the somewhat costly as compared to the others, so we are trying to reduce the quantity of the silver nanoparticles. That's why we have prepared a different ppm solution, and we are putting the 10 larvae in each beaker, and we see that the mortality rate is increases as per the graph. These are the XRD analysis of the synthesized material. As we can see that the particle size is near about the 70 nanometer, which was confirmed with the help of XRD and the particle size analysis. These are the thermal gravimetric analysis. As we are already discussed that our particle is stable up to the around the 450 degrees Celsius, so it having the good thermal stability. These are the elemental analysis. These are the antimicrobial activity. We are done the antimicrobial activity with the help of microbiologists, and as we can see that it is more effective towards the gram-positive bacteria as compared to the gram-negative bacteria. Finally, we are concluded that We are synthesizing the silver nanoparticle according to the green chemistry, which is the economical, efficient, and the eco-friendly, which are characterized by the different characterization techniques such as FTR, PG, FSM, XRD, and other. And this is the eco-friendly process, and this technique can be employed into the society, and still we are working on that. These are the acknowledge. I'm very much thankful to the our coordinator, principal, my guide, Professor J.S. Meshram, then our co-guide, Dr. Vikas Gite, all teaching, non-teaching staff, and all our organizers from the your college. Thank you very
Thank you,
much.
sir, for giving me an opportunity. I am Dr. Navdeep Sharma, Assistant Professor, Department of Physics, School of Basic Sciences, Avidas University, Mandi, Himachal Pradesh. In this second international conference on recent trend in material science and devices 2023. My research work is on electrical charge transport exploration of camphor sulfonic acid doped polyorthomethyl aniline is a conducting polymer. So before to start my work, I want to discuss which I want to present in this. First is the introduction about the conducting polymers, and second, synthesis methodology of the polyorthomethyl anilines, a conducting polymer, and the characterization technique which I have been used for studying polyorthomethyl aniline is FTIR, UV visual spectroscopy analysis, XRD analysis, and the main is electrical charge transport exploration models, which have been used for studying the polyorthomethyl anilines and their conclusion and future scopes. First, I want to discuss with you that this conducting polymer is a another and very important type of smart materials, and that was first discovered by Alan J. McDermott, Hideki, Sirikawa, and Alan J. Higer. Sir, am I audible? Hello.
Yes, sir, you're audible.
Okay. Thank you, sir. So the conducting polymer is basically called intrinsically conducting polymer, is a organic polymer that conduct electricity, and such compound may have metallic conductivity or can be semiconductors. These conducting polymer are special one or called be as smart polymers. Unlike traditional polymer that are insulators, conducting polymer possess electrical conductivity, allowing them to conduct electricity. Basically, the conducting polymers have such very unique property due to their delocalization of electron within their molecule structure, and that have a very tremendous application in electronic devices and semiconductor technology and in medical science too. So next, the conductivity of the chart, it shows that the conjugated polymers have shown their conductivity in the semiconductor range or a metallic range, silicon, germanium, or even the copper, iron, or silver too. And that's why the conducting polymers are more demanding in electronic devices or semiconductor technology, even in the medical sciences too. Next is the chemical synthesis method for polyorthomethyl anilines. That is the methods. There are so many methods to be used in for the formation of conducting polymer in electrochemical methods or another methods also. But here I am discussing a very cheap and very easiest method, that is a chemical oxidative polymerization method for the preparation of polyorthomethyl anilines. The process involved basically a monomer orthomethyl anilines that we use in distilled water at a low temperature at 0 to 5 degrees Celsius, and the solution was then added into 30 millimoles dopant, that is a camphor sulfonic acid, in 50 ml of distilled water. When after the proper mixing of the monomer and dopant, we started the polymerization process. And for the polymerization process, there is a need, we use here in our process ammonium persulfate as an oxidizing agent, and in very gentle way, drawwise additions of the ammonium persulfate as an oxidizing agent in the solutions, the polymerization process starts. Basically this ammonium persulfate is also pre-cooled at 0 to 5 degrees Celsius to maintain the reaction temperature, because if we not maintain the temperatures, then there is a possibility of secondary polymerization process, which were not to be useful for the desired conducting polymers. As the polymerization process is completed, the reaction durations is required for that one, for the completion of the polymerization in a complete way. For that, the reaction process is carried out for 7 hours at room temperature at the magnetic stirrer. And after the completion of the polymerization process, the materials that is obtained is filtered with Buttman filter paper 42 and washing too many times to remove the impurity by using distilled waters. After the completion of the filtering process, the material is to be drying in vacuum ovens for further to remove the moisture from the conducting polymers. And the process is to take a long time, 24 hours in the vacuum ovens. And after the completion of that, once we have obtained a green colored polyorthomethyl polymer that is shown in the figure, that gives the first indication that our conducting polymer is prepared. And after this, we have used a mortar pestle and make it for the further process, and we have prepared a pellets of the polymers for the further studying with the help of a hydraulic press. This is a pellets that is we have used for further study in the electrical conductivity and the charge transport mechanism of the polyorthomethyl aniline conducting polymers. There are basically this polyorthomethyl aniline have a different oxidation states. That is, one is licoamidine state as shown in the figure 1, and amidine base and conducting amidine state, and fourth one is perniciline state. Basically, these states are to be is the rich chemistry of this polyorthomethyl aniline conducting polymers, and the prominent one state of this is a conducting amidine state, which is responsible for the conductivity behavior in the this conducting polymers. And here you have seen that CS3 group, that is a methyl group, is to be attached at this ortho positions of the polymer chains in this long chain polymers. And we have further for the knowing whether the dopant camphor sulfonic acid is attached in the this monomers or not, to be studied with the help of FTIR analysis and UV visual and XRD study. And here we have seen that the first one is a black color. This is a D-doped polymers which we have shown different peak, and the rest other states of the red and others ones, we have optimized samples shown that different peaks, and the peaks that is obtained in doped samples is a prominent peak, that is 10111, confirming the polymer formations, and this one indicating the non-symmetrical CS ring stretching modes. That confirm that the our CSA dopant is attached within the orthomethyl positions of the conducting polymers, and the band around 3400 and 3200 wavelength broadened compared to the D-doped amidine base, which show the involvement of amine and imine and NH groups, and have to give the confirmation that in doped state this presence gives the confirmation that the dopant material is properly attached at the ortho positions as compared to the D-doped state. The appearance of the peak at 1742 wavelength signifies carbon oxygen stretching vibrations, indicating CSA ion is incorporated in the polymer backbones. The red shift from the approximately 1588 to 1533 wavelength and from 1500 to 485 wavelength suggests the amiridine base conversion, that is a amiridine conducting state to the semiconide forms, confirming the protonations which is responsible for the conductivity in the polymers. The distinctive peak at 2958 wavelength resulting from the CH vibrations remarkably influenced by CH3 and CH2 groups from camphor sulfonic acid dopant. The peak at 134.49 wavelength for doped state represent aromatic CN stretching, indicating a secondary aromatic ion groups, while 615 wavelength peak signifies CH out of plane bending vibrations of the CSA groups. And the UV visual spectra of polyorthomethyl anilines for the dedoped and others samples S1, S2, S3, S5 sample. Actually, these S1, S2, S3, S4, and S5 sample is the dopant to monomer concentration. We have increased the concentration of dopant with respect to monomers for sample S1, S2, S3, S4, and S5 with the ratio 1, 2, 3, 4, and 5. And we have to check whether at which dopant concentration our conducting polymer shows better results. And the SAR peak at 320 nanometers shows the pi-pi star transition for the benzonoid rings, and the very small peak at 420 nanometer for localized polarons, which is the key factor for the conductivity in the polymers. The soldiers at nearly 610 nanometers, indicating the insulating pernigrine segment, and the shift in the wavelength after doping signify a dual state in the polymer, that is a conducting, and revealing that that in comparison to de-doped state, there is a formation of the conducting polymers in the proper way. This is also confirmed with the help of our XRD study of polyorthomethyl anilines. The pattern, as you have seen here, that de-doped is a black one, shows and there is a no peak at these positions. What
Dr. Navdeep, please wind up.
Okay, sir. So the peak and broad diffraction peak at 15 and 16 degree or 24 degree indicate that semi-crystalline or amorphous characteristic in the existing literatures. And the electrical conductivity measurements by using the 4-pro methods, by using this sample that IV study represent that the maximum conductivity of the sample to be shown at 1 into 10⁷ per centimeter. For the best, it shows that the sample ratio 2 shows a best conductivity for the samples. And the temperature dependent conductivity is done at 300 to 430 Kelvin. The conductivity shows the exponential behavior, and it shows that the conductivity is in exponential behaviors, and this behavior is very useful for the further study of electrical charge transport exploration of the polyorthomethyl aniline, that how that conductivity in the polymer orthomethyl aniline is going on with by moving the charge carrier in them. There are Arrhenius model, very important model that is to be used for comprehend the charge transport behavior in the polymers. And this is a formula that is sigma dc sigma naught exponential minus E upon kVt is used here, where EA is the activation energy and kV is the Boltzmann constant, is the temperature, sigma naught is the pre-exponential factor that is used to explain the charge transport behavior in this one. Activation energy is very important here because it gives us the idea that whether the charge carrier is moving in the polymer chain properly or not. And here the Arrhenius model used for this polyorthomethyl CSA, and you have seen that this have shown that for the sample S1, S2, S3, S4, S5, and here the conductivity range is 350 to 300 to 430 kelvins, and after 430 kelvin, that we have seen that the line is not fitting well. It means that this model is applicable between the range of 350 to 430 kelvins, and the data that is obtained from this one is activation energy. That is very important to understand that whether the charge is moving properly or not. And the activation energy for the sample 2 is found to be very less, that is 308.308 as compared to the other sample. So this 0.308 activation energy is less in this case. It means that the conductivity is found to be better in this sample S2 ratio, because the charge required less hopping for this ones. And another is very important model, that is mode variable range hopping models, and this model is used in one-dimensional, two-dimensional, and three-dimensional, as the formula is given here, sigma naught exponential minus t naught upon t upon 1 upon n plus 1. Here n is the for first, second, and third dimensional model, and we have to check our conducting polymers whether this model is applicable for this charge transport model or not. And we have done this one by using the different parameter t naught, and this is a average hopping distance and average hopping energy. And we have explored that for one-dimensional, two-dimensional, that our conducting polymer is well applicable at 300 to 370 Kelvin temperature, and below that the line is not fitting well. But in case of two-dimensional, that fitting is well at 380 Kelvin, and after that it is not find well. And it means that it is applicable up to this temperature, within these temperature range. And for the three-dimensional VRH model, the temperature range is 300 to 420 Kelvin, and this one is shows that 420 Kelvin temperatures the model is fitting well, and the charge transport behavior is showing better in up to this, and not beyond this ones. So it means that in case of Arrhenius model, the conductivity range is found to be at 350 to 430 Kelvin, but in case of one-dimensional mode variable, the temperature is 300 to 370 Kelvin, but in two-dimensional 300 to 380 Kelvin, and three-dimensional it is found to be 300 to 420 Kelvin. It means that in case of mode variable range model, the conductivity of the charge transport is in long temperature range, but in case of Arrhenius model, it shows that the temperature of the charge carrier is found fit at a higher temperature. So in case of our conducting polymer, polyorthomethyl aniline, we found that there are different parameters that is obtained from mode BH model applicability, that the conductivity found to be better in case of sample 1, that is 1 into 10⁻⁷ Simon per centimeter, and you have seen that the variable hopping distance is 7.6 and hopping 301. It means that hopping distance is less, then hopping distance for the charge carrier is found to be less, and the energy density of state that is obtained in this ratio S2 is maximum as compared to other samples. It means that
Please, Dr. Nabdi, please conclude.
Okay, sir. And we found that in case of the room temperature conductivity of this our sample is 1.0 into 10⁻⁷ Simon per centimeter observed at CSA to monomer ratio 3, and the charge XRD confirmed dopant presence in the doped state, and FTR confirmed the successful polymerization and identified the characteristic ring vibration shifter. And the charge transport study through Arrhenius and Mott models revealing mixed mechanism in polyorthomethyl aniline. It means that all models explain inclusively the charge transport behavior to in the orthomethyl anilines conducting polymer, and this polyorthomethyl aniline conducting polymer have a very advanced technology in sensor, energy storage device, optoelectronic device, and in medical science too. We are also working on the sensor behavior of polyorthomethyl anilines in the coming days. Thank you, sir, for the
Thank you, the organizers, for giving me the opportunity to present my work. So It is Kelvin probe force microscopy study on antimicrobial activity or by graphene oxide nanohybrid. So this is the work that I did during my postdoctoral fellowship in IIT Guwahati. So presently I am working as an assistant professor in Department of Physics, Sipazar College. So it is in Assam under Guwahati University. So my talk will be categorized in this section like introduction. So I will briefly discuss what is the Kelvin proof force microscopy, and then the experimental background, that is how I have made the composite, and what is the actually the results I have got, that will be discussed in the characterization part, and then discussion and the conclusions and the references that I have used, and then acknowledgement. So introduction part. So basically, this has shown about the graphene. So what is the graphene look like? So everybody knows it's a honeycomb structure. So 6 carbon atoms are there in the single sheet of graphene. So it's if I say about the unit cell, so each unit cell of graphene that contains 2 atoms of carbon. So it is shown here like this 2 gray and green carbon atom, they form the motifs, and the overall crystal is given by the lattice and the motif structures. But the unit cell is basically the structures that has been given by this rhombus. Okay, so what are the different uses of graphene? So graphene basically, so if you see in the right hand side, so graphene, there are lots of applications in the graphene. So basically during my work, I basically worked on the semiconductor property and the optics property. So here in this presentation, I will show something regarding the semiconductor property of the graphene and the composite material. So overall, what I am going to show is how the bacteria basically dies when it's come in contact with the graphene material. So I have chosen the graphene composite, which is metal phthalocyanine metal phthalocyanine material, which is zinc phthalocyanine I have chosen, and it is basically a good electron withdrawing agent. And if you see the bacteria I have chosen as the Escherichia coli, that is E. coli, which has a lipopolysaccharide membrane, which is an electron efficient membrane. So what the ZNPC basically, it withdraws the electron from the graphene, and then the electron which releases from the bacteria cell, that electron withdrawing power of the graphene will basically lead to the death of the bacterial cell, and that is basically detected with the help of this Kelvin probe force microscopy, which I am going to discuss basically what is the electrical property, how it can be actually measure the electrical properties by this Kelvin probe force microscopy. So this is basically actually advantage which is gained from the AFM. Actually, what we see in the AFM, we use a metal tip. Okay, so in the case of a metal tip, actually it is when it comes in contact with the semiconductor-like surfaces, so it forms a ohmic actually structures where there is a change of this band gap and the band alignment occurs. So if we give the compensating voltage from the outside, then we can basically measure what sort of a voltage dissimulation is going on in between the tip and the semiconductor surface, and that is basically calculated with the help of the change of the work function between this surface and the tip. So it's the equation that is used to calculate this contact potential difference. This is the VCPD, which is the work function of the surface minus work function of the tip divided by electron. So for our case, we keep this electron charge as 1. So this is the schematic of the KPFM. So what it does, basically, this is the metal tip. So this has a work function of phi t, and if you see here, it is the work function of the sample, semiconducting sample. So there, if they comes in contact, so there is a band alignment that is going on. So there is a appearance of this contact potential difference. So if we give some potential from the outside, so this potential will compensate this VCPD this contact potential difference, and from that actually the instrument calculates the whatever the change in the voltage that is occurring over your surface. So this is the actually image that I have taken from the broker. So they have shown here in a very good way how one surface can be imaged, and at the same time you can also image the surface potential, which you can say the mapping or the stepwise. Like if you have different steps with different voltages, that will give you the different voltage map in terms of the change of the voltage. So this is the phenomenon, this is the mechanism actually that I have used in order to sense the contact potential difference between the surface and the tip, which alternatively gives me the change during change that is occurring during the date or during the electrical charge assimilation that is going on between the bacterial cell and the this composite material that I am using. So the experimental background that I have used, so I have actually deposited this metal phthalocyanine that which I have used as zinc phthalocyanine, so that is coated over a ITO substrate. So at around 350 degree centigrade by thermal evaporator method, and then graphene oxide material, so that is deposited over the zinc phthalocyanine by a homemade dripping method. So that is actually I used during my process. So this composite thin film materials were heated so that we can get the ZNPC, which has the structures of J aggregate and H aggregate. So the heating basically changes the structures, which I will discuss during my results part. And then we did the characterization part in order to actually see whether the interactions between the graphene and the ZNPC layer basically gives us the good assimilation or the good composite formation. Then we did the electrical measurements and the KFM measurements in order to get the death mechanism of the bacterial cell over our composite material. So this is basically the characterization part. So we did the FECM part. So FECM, you could see that in figure A, this is the as deposited ZNPC film, and this is basically in figure B. We have shown here the ZNPC film when it is heated to a temperature of around 585 degrees centigrade. So this is the change of the morphology where the J aggregate changed from the H to the H aggregate. So this is the structures of C shows the J aggregate structures when graphene oxide is deposited over them, and D shows the same material, the same composite material when it is heated to 585 degrees centigrade. Okay, so we did the XPS study in order to see the assimilation between these two layers. So if you can see here, there is a peculiar peak here at around 285. So it's actually showing that that is the interaction of the carbon and the nitrogen, and the other peaks are carbon oxygen in different valency range, and the carbon interaction. So the carbon nitrogen peak you can see in the figure D, it is highly increased. So from there we can see that this the interactions between the graphene and the ZNPC peak has shown a very good actually promise. So the other peaks like the carbon oxygen interactions in different valencies and the carbon interactions, they have been diminished. So from here we can see that the interactions is increased when we use basically the heating for our composite material. So this is also shown here, the Raman graph and the Raman mapping. So here we have convoluted the Raman graph from this red graph. This is the heated graph, actually the composite which was heated to 580. So it is shown here that two peaks at around 1300. This peak and this peak is for the graphene oxide, and the other peaks are for the phthalocyanine. We had to convolute this composite peak in order to get in order to show the different interactions between this composite material. We also did the mapping, Raman mapping for our composite material, and from here we see that when it is heated, the interactions is proper. So the graphene oxide and the different parts of the composite material is aroused. So in case of non-eating, where this ZNPC in a J aggregate form, so their interactions is very less. So from from this characterization also, we could see that the interactions between the two molecules were very prominent. So we did also the AFM topography and phase characterization. So where we can see that the interactions between the graphene oxide and the ZNPC were very high when it is heated. Actually, the naming AB, so they have been for A is for your topography and B is for your phase. So phase basically gives the interactions, mechanical interactions between the two composite layers. So you can see when it is in a J aggregate, they don't have very distinctive interactions, but when it is in H aggregate forms, they have a very distinctive interactions, which actually ascertain the good interactions between these two composite material. We did the electrical characterizations also and the electrical mapping actually with the help of this AFM tip. So this shows that the prominent interaction sites were arousing when we did the heating of our composite material. So this shows that this material could be very efficiently used for doing different electron withdrawing or electron changing or charge assimilation calculations or charge assimilations applications. Okay, so this is the work actually when bacterial cells were deposited over our composite films. We could see that when it is in J aggregate form, they were live, but when we the bacterial cells were deposited over the composite material which were heated to 585 degrees centigrade, so in the H aggregate form, so there were very less number of live bacterial cells, and from there we actually did the electrical measurements, this KPFM measurements. So KPFM measurements were before the incubation, like when the composite material were in the J aggregate form, they did not show any sort of change even if we do the characterization for a longer time. But when we did the incubation, that is for our H aggregate composite material, so we could see that the bacterial cell, when it get incorporated, this is the topography. So over the this graphene and the bacterial cells, so we could see that there is a charge assimilation as the time passes. So this shows that now this is at around 0 minute, means at the initial part. So the all the charge, this yellow mapping color is basically the charge that is possessing by the this bacterial cell, which is also shown here before the incubation. So as the time period changes, basically this yellow color, basically the charge which is shown by the yellow map, so they have been starting to assimilate with the surface, which is the ZNPC surface. So after a long period, at around 1 hour,
Hello,
1 hour time, you could see all the charges that
hello,
has.
Dr. Mani.
Yeah, sure, sir.
Please conclude.
Yeah, sure, sir. So I'm concluding. So all the charges that has been come to the surface of our surface of the composite material. So that all the calculations using the equations that has been provided earlier has been shown here, and we could see that a cell level charge assimilation during the death of the bacterial cell can be shown with the help of the KPFM. So we have here given a mechanism how the electrical charge that has been assimilated and with the composite material, and this lead to the death of the bacterial cell. So these are some of the concluding remarks which have already been discussed during my talk. And I would like to acknowledge the CI facility at IIT Guwahati, then the Center of Nanotechnology IIT Guwahati, and my supervisor or mentor during my postdoctoral fellowship, Dr. Dipankar Bandyopadhyay, the Chemical Engineering Department IIT Guwahati. Also acknowledge the facilities in SINP Kolkata and my lab mates during IIT. And also I want to acknowledge my college where I have been employed right now. So thank you very much.